Okay, well, thank you very much. I'm really sorry that I could not be here in person with you due to some unforeseen passport requirements, but at least thanks to the technology, I can be here in spirit. And thank you very much for attending this talk. I have to confess that this title is not my own. This title was suggested to me by Rod Eckenhop, who also could not be here, but for very different reasons. But I thought I should come up with my own title. Please, next slide.
2.
Yeah, so my real title is Waking Up from Anesthesia, Harder Than Making Flippy Floppy. And that, of course, is a reference to the Talking Heads. Next slide, please. And here is David Byrne and his glory, but you can also, will realize in a couple of slides why I think this title is much more relevant. Next slide, please.
3.
I'm going to start by telling you things that I think you all know very well, and that is when we think about pharmacology, typically think about is the relationship between the drug concentration and its effect observed in a population of patients or animals or whatever it is we're studying. So when we think about anesthetics, the concept of MAC has been very influential, and originally MAC was defined as a concentration at which 50% of people lose their ability to respond to a surgical stimulus. We can define other anesthetic endpoints, like for instance, MAC awake would be the concentration at which half of the people lose their ability to respond to a verbal command. Next slide, please. Of course, when we're talking about animals, it is very hard to study their responses to commands. So instead, in rodents, typically we think about the righting reflex, and I will tell you a lot more about it in a moment.
Alex, is this slide 3 or 4?
No, we're on the right slide. Perfect.
Okay.
The only thing I want to say about the righting reflex at this point is that for a variety of anesthetics, the concentration at which mice lose their ability to right themselves is very closely correlated with the probability, with the concentration at which humans lose their ability to respond to commands. So in rodents, we can use the righting reflex as a proxy for being awake. Next slide, please. Okay, so I want to just tell you why we're doing the things that I'll tell you about next, and that is all the measures of anesthetic effects that I have described so far are defined for a population of people. But if we really want to develop personalized medicine and personalized pharmacology, what we really want to know is not the average response in a population, but the response to anesthetics, in this particular case, for each individual. And as that's what we're trying to do here. And as you will see, it's not as simple as one might originally. Next slide, please. So what is the righting reflex? So in our lab, we have this apparatus where we place mice in these little cylindrical chambers, and we can flow in an anesthetic into these chambers. We can measure anesthetic concentration in each one of these chambers, and we can control the situation rather precisely. So a righting reflex is tested by rotating this chamber 180 degrees, such that the mouse ends up on its back, and we simply determine whether a mouse is able to right itself. So say, for instance, I dialed my anesthetic concentration to a MAC for righting reflex. And I do this experiment on 2 different mice that are marked individual 1 and individual 2. I would expect that one mouse, individual 1 for instance, would be able to right itself, but individual 2 would not be. And that is the point. At the level of each individual, the responses are binary. A mouse either is able to right itself or it isn't. A human is either able to respond to a command or they're not. And most importantly, if I repeated this experiment, next slide, please, I would expect that the responses be consistent across time at a fixed drug concentration. So if a mouse 1 was able to right itself before, it should continue to be able to right itself next, and same for individual 2. Previous slide. I wasn't quite done. Yeah, and the whole point is that you get binary responses of individuals, and to get smooth population-level responses, what you need is variation between individuals. So for instance, mouse 2 is a bit more sensitive to anesthetics than mouse 1, and that's why at the same anesthetic concentration, it stays on its back, it's not able to right itself, but mouse number 1 is. Next slide, please. So here we decided to test this assumption. And what I'm showing you here are data from 20 adult mice that are all siblings. They're highly genetically related to each other. We exposed them to anesthetic concentration, in this case isoflurane, for 2 hours to equilibrate them with the concentration, which is much longer than it takes for pharmacokinetic equilibration. And what I'm showing you here are responses of each individual mouse to repeated trials of the Righting reflex, which we administer every 3 minutes for 40 times. And what you can see is that for every individual mouse, the ability to write itself fluctuates. It flips and flops. Therefore, my title. So if a mouse was able to write itself here, it gets a green square, and if it was not, it gets a blue square. So you can see while individuals fluctuate in their ability to respond at the level of population shown below the plot, you can see that the population level response stays constant at roughly about 50% likelihood. Okay, so this is this kind of a flipping and flopping that is quite unexpected at a fixed anesthetic concentration. Next slide. So the simplest possible model of these fluctuations is essentially a coin toss. Let's imagine that while the population is at EC50, that this has 50% probability of writing, each animal is also at EC50. That is, it has 50% probability of writing itself on each trial. So this is equivalent to tossing a coin on each trial, and if the coin lands on heads, then the mouse is able to write itself, and otherwise it is not. The key assumption of this model is that the responses on the next trial should be entirely independent of the previous one, because my next coin toss does not depend on the previous coin toss. So we thought we should test this assumption in the data. Next slide, please. So simply here we have compared the probability of a Righting reflex in two different situations. In the first situation shown by the gray box plot, we looked at all mice that were found to be awake on the previous trial, and we looked at their probability of becoming unresponsive on the next. And in the pink box plot, we're comparing this probability to those mice that were found to be unresponsive on the previous trial. And what we can see is that the probability of unresponsiveness in this case is much higher. And the point of this is that the response of each individual depends not just on the drug concentration, but also on their individual previous history. Next slide, please. Okay, so because this was quite surprising and we were able to replicate this experiment now in hundreds of mice for every volatile anesthetic that we could get our hands on, we thought we should try a different model organism to see if this fluctuation is specific to volatile anesthetics or to mice. So for this purpose, we used a larval zebrafish, which is a very well-known model system for studying genetics, but also for study of pharmacology. So in zebrafish, we cannot test Righting reflex. Instead, we use something called a startle reflex. And that is if a cuvette where a fish is situated is gently tapped, the fish will exhibit these stereotyped escape behaviors that you see on the bottom left. And we can image those using a high-speed camera. So what you're seeing on the blue and green plot on the right is an analogous experiment performed in 72 zebrafish, which are each bathed in a 3 micromolar propofol, which happens to be about 50% effective concentration for loss of startle reflex. And what you can see that in every fish, just like in every mouse, the ability to exhibit startle reflex fluctuates from trial to trial, although propofol concentration stays exactly the same for the duration of this experiment. And in this case, we're showing you 360 trials of startle reflex administered every 15 seconds. Next slide, please. Just like in the mouse, in the fish, the ability to exhibit startle reflex depends on whether each individual fish was responsive on the previous trial, that's the gray box plot, or unresponsive on the previous trial, and that is the pink box plot. Next slide, please. So this leads us to a remarkable conclusion, is that while a concept like drug potency is extremely useful for describing responses in a population, it is not sufficient to describe responses in each individual. And that is because effective concentration does not take into account what happened to each individual before. And the responses in each individual depend not just on the drug concentration, but also on their previous history. So that's a fundamentally novel insight that comes from studying responses in individuals rather than in a population. Next slide, please. So this, of course, is very reminiscent to what is known in molecular biophysics. So let me give you an example, okay? The plot on the left shows a dose response for a current elicited by a GABA A receptor as a function of concentration of GABA, which is shown on the X. So this recording is done by recording currents flowing through the whole cell that contains many GABA A receptors. But in the plot on the right, what the authors were able to do is to record a single GABA A channel. This is a technique that has been developed some 30 years ago and has been applied at this point to just about every ligand-gated and voltage-gated ion channel. So what you can see that while the concentration response curve for a population is a smooth and graded function, ion channel exhibits fluctuations between the conducting and the non-conducting state. This is true for every voltage-gated and ligand-gated ion channel. And what you can also see is that once a particular state is established, it tends to persist for some time, which is defined by its characteristic time scale. So next slide, please. There is a very sophisticated mathematical apparatus that has been defined for these fluctuations in ion channels, and I will not go into the details of the math here, but suffice it to say that it involves modeling transitions between different states. So let me explain what I mean. Let's imagine that my mouse is in the responsive state, shown by the green circle. Then there are two probabilities. One possibility is that on the next trial, the mouse will stay in its previously observed state. That's this sort of self-looping connection. The other possibility is that the mouse will switch its state. Okay, and I can define similar quantities for the unresponsive state. Okay, and next slide, please. Okay, and the key insight here is that the sum of all probabilities leaving a state must add up to 1. So while there are 4 arrows in this plot, all I need to know are 2 parameters, one for staying in each state, one probability for staying in the responsive state, and another for staying in the unresponsive state. Next slide, please. And why would we want to do this? Well, because it gives us the ability to quantify not just drug potency, but also a novel quantity here that we will call resistance to state transitions. So consider 2 different individuals that have been exposed to repeated Righting Reflex trials. Both individuals, in this case, have the same probability of writing overall, 50%, just for the sake of illustration. But note, individual 1 does not switch their state very frequently. They tend to stay where they were for longer than individual 2. And therefore, the number of state transitions in the individual 1 is much fewer than those observed in individual 2. So by looking at transition probabilities, we can estimate not just the overall probability of writing in this case, but also this degree of resistance to state transitions. Next slide, please. And both of these properties, the resistance to state transitions and the overall potency of the drug, are reflected in a parameter space that is spanned by the 2 parameters that we need to estimate the system. That is, on the X, I'm showing you the probability of staying in the unresponsive state, and on the Y, I'm showing you the probability of staying in the responsive state. And each point in this plane is colored by the overall probability of writing. If the plot is blue, that means there's a very low overall probability of writing, and if it is green, high probability of writing. But notice that many different combinations of parameters result in the same overall potency. Specifically, if you move anywhere along a black dashed line, the potency will not change. Individual 1 and individual 2 from the previous slide would be found along the same lines. Whereas resistance to state transitions is defined by staying along the red dashed line. Anywhere I go along the red dashed line, I will change the probability of writing, but the probability of state transitions will stay approximately the same. So in this case, evaluate both the similarity between individual 1 and individual 2 in terms of their overall sensitivity to anesthetics, but also at the same time quantify the differences in their resistance to state transitions. Next slide, please. Okay, so if we were modeling an ion channel, we would combine many experiments in different individual ion channels into one model. But when we looked at the data from mice, we realized that that may not be such a great idea. Next slide. So these are just a replotting of the data from a couple of slides back, and I just want to illustrate something for you. So this particular mouse was able to write itself on 3 out of 40 trials of the writing reflex. Next slide. Whereas its sibling, exposed to the same anesthetic concentration in exactly the same experiment, just in a different anesthetic chamber, was able to write itself 34 out of 40 times. So this is an order of magnitude difference between two individuals that are ostensibly genetically identical, but nevertheless, they exhibit dramatically different overall probability of writing. So therefore, what we decided to do is, rather than model the whole population of mice, is to come up with a model for each mouse individually by computing its overall transition probabilities between states of responsiveness and unresponsiveness. Next slide, please. So therefore, in our plot that represents the probability of staying in the unresponsive state on the X and probability of staying in the responsive state on the Y, each mouse is represented by a point. So what I want you to see is that individuals are indeed different in terms of the parameters of the model that describe their fluctuations at a fixed anesthetic concentration. Some animals are much more sensitive. Those are shown by the blue points. Other ones are a bit more resistant in that they write themselves on more trials.
Can you maybe restart the slide?
But the variability amongst individual mice is highly structured in that they're not just randomly distributed in the plane spanned by the two transition probabilities. They're found predominantly along the line. And recall from the previous slide that this is the direction along which the resistance to state transitions is conserved. So whereas different individuals can differ dramatically in terms of their sensitivity to anesthetics, this novel property called resistance to state transitions appears to be largely conserved amongst them. Next slide, please.
So we're going to slide 18 now.
Yeah, that's right. We're on the right slide.
Good.
Yeah. Yeah. So this is the same kind of analysis for the zebrafish now exposed to propofol. And remarkably, we see the exact same thing happening. Different individual zebrafish can differ dramatically in terms of their sensitivity to 3 micromolar propofol, measured by the overall probability of startle reflex. But the variability amongst individuals is predominantly found along this line that conserves resistance to state transitions. And I would like to point out that this is remarkable because fish and mice are separated by about 400 million years of vertebrate evolution. Next slide, please. So I just want to summarize the last couple of slides and to say that by studying individuals' responses to anesthetics, we can appreciate that even very similar individuals can exhibit dramatically different sensitivity to the drug. And this novel measure called resistance to state transitions is conserved amongst them. So while animals fluctuate, flip and flop between states of responsiveness and unresponsiveness at a fixed drug concentration, the probability of switching between states appears to be conserved. Next slide, please. So then I want to bring this back to this concept of neural inertia, which was first introduced by my good friend and collaborator, Max Kells. So what Max has observed is that if you compute a dose response curve for induction of anesthesia, you will get the right-shifted dose response curve on the right. However, if you take the same mice and you try to produce the same kind of dose response curve for emergence from anesthesia, the emergence curve is shifted to the left. That is, it takes less drug to induce anesthesia than it does to wake up from anesthesia. And he termed the difference between these curves as neural inertia, which expresses this resistance to transitions between states of wakefulness and states of unresponsiveness. I should also say that some human studies have supported this concept of neural inertia, whereas others have not. So it is not clear whether this exists in humans, but it has been shown to exist in animals ranging from Drosophila, a fruit fly, to mice. Human work is more complicated in that it is very difficult to measure the anesthetic concentration in various body tissues, whereas in mice we have done experiments to confirm this anesthetic hysteresis by measuring concentration of anesthetic in the brains of animals. So it is controversial on a human level, but in animal models, this phenomenon definitely exists. And the question arises, can the fluctuations between states of responsiveness and unresponsiveness in each individual give rise to this neural inertia observed at the level of population? Next slide, please. So to address this question, Drew Hudson and I studied the process of switching between responsive and unresponsive state in a mathematical model. And I won't bore you with the details of the model, but suffice it to say, our conclusions were that yes, indeed, this state switching can give rise to hysteresis, but there is a novel prediction based on this model, and that is the hysteresis should collapse. So what you're seeing in the green curves is if the drug concentration is changed rather quickly, then there is profound hysteresis. But if you wait longer and longer and longer at each anesthetic concentration, then the hysteresis collapses, and that is shown by these cyan curves. So what determines this time scale of collapse? Well, in the model, it is determined by the amount of noise. The noisier the system, the more rapidly it switches between two states, the more rapidly the system equilibrates and the hysteresis collapses. The less noise, the longer the system tends to dwell in its previously observed state, and the longer the hysteresis lasts. Of course, in the model, it's very simple to control anesthetic concentration. This is not so simple in animals, where pharmacokinetics would also contribute to hysteresis. But nevertheless, the model predicts that if this equilibration time is longer than the pharmacokinetic equilibration time, we should observe hysteresis collapsing as a function of noise rather than function of pharmacokinetics alone. So we thought we would test this prediction of the model in the data. Next slide. And for this purpose, we performed a pretty simple experiment. We exposed mice to 1.2% isoflurane, at which point all the mice are reliably unresponsive. They're not able to right themselves on any trial. And we compared the same mice in a different setting in a following paradigm. Next slide, please. Instead of exposing them to 1.2% isoflurane, we exposed them to no isoflurane at all for half an hour, and then we switched to 0.6% isoflurane. So this is our model of induction and emergence, respectively. And what we wanted to see is how does the population level writing probability evolve in time during this paradigm. Next slide, please. And what we can see is exactly consistent with the predictions of the model. So on the yellow curve, what you're seeing is the induction. That is, an animal was exposed for 30 minutes to no isoflurane, and the time starts when 0.6% isoflurane is turned on. And what you can see is that at the level of a population, the writing probability evolves very slowly and eventually comes to a steady state. When we do the same experiment after exposing the mice to 1.2% isoflurane, the same kind of thing happens. And I hope you can see that these two curves come together. Now, and I'm not showing you this data, but this is essentially a replication of previous work. It takes about 10 minutes for mouse brain to equilibrate with isoflurane concentration. And that point is shown by the white dashed line. Notice that the time here is logarithmic, so it takes roughly an order of magnitude longer for hysteresis to collapse than it does for the drug to equilibrate with the brain of an animal. Next slide, please. So I just want to summarize my talk in its entirety and to say that what we have done here is try to define responses to anesthetics in each animal individually. And when we do this, we realize that even when the concentration of anesthetic is fixed, each animal fluctuates between states of responsiveness and unresponsiveness. These fluctuations exhibit inertia. The animal is more likely to stay in its previously observed state than it is to switch states. This neural inertia is conserved amongst animals from zebrafish to mice. And furthermore, we have shown that this kind of fluctuation can give rise to neural inertia observed in previous work. And furthermore, we tested a prediction that this hysteresis should collapse. We were able to confirm that in experiments. And this hysteresis collapses on a time scale which is much slower than pharmacokinetics. Next slide, please. So I just want to leave you with this great quote from a famous British pharmacologist called David Calhoun at UCL. And when he was saying this, he talked about single receptor molecules. And he said that the actual microscopic constants, that is the constants that define the switching between the responsive and the unresponsive state in our case, tell us about what is going on. In contrast, concentration response curve defines a macroscopic constant. It describes what we see, but it does not tell us what is going on underneath. And I think that this is, while this is talking about ion channel, this is quite apropos for individual animals. That is, when we observe the concentration response curve, we would have no idea that each animal actually fluctuates between states of responsiveness and unresponsiveness, and that this pattern of switching expresses a highly conserved pattern. Next slide, please. This is perhaps the most important slide. I want to thank all the people that contributed to this work, most importantly, Max Kells, who has started the study of neural inertia and has been my friend and collaborator for years now. Andy Vasilchev, Andrew McKinstry-Wu, Ben, and Mfangi have all done these rather tedious experiments of flipping mice for hours on end. And Vicky Bidell is an investigator at Penn with whom we collaborated to do experiments on zebrafish. I want to thank my funding sources, which is NIGMS and John S. McDonnell Foundation and the Department of Anesthesiology. Next slide, please. And of course, I want to thank you very much for allowing me to give this talk via Skype. I'm sorry if I was a bit delirious. It's 4 in the morning here, and I'm not quite awake myself. And I'd be happy to take any questions.
Alex. Thank you so much.